Hey everybody, sorry for the delay in starting. I was caught up in a conversation about large language models. So we're going to start this tutorial workshop session. So just make sure you're in the right room. This is the Banksy Unifying Cell Typing and Tissue Domain Segmentation for Scalable Spatial Omics Data Analysis by Vupal Singhal. And with that, I'll let you jump in, and sorry for taking up time.
Hello. Hey guys, thanks for being here. This is package demo. So the way we'll do this is in two parts. I'll go over the core concepts of Banksy and the use cases and show you guys some results. And then Joseph will take over, and he wrote a lot of the Bioconductor code. He incorporated, like originally Banksy was its own Banksy S4 object, but then Joseph turned it into Bioconductor compatible spatial experiment objects and so on. It's really great work. So he's gonna go through the code. Okay. Okay, yeah, so Banksy at a very high level, I think it's, even though we've talked about it as a domain segmentation and cell typing method for spatial data, it's actually a little more general than that. It's sort of incorporating spatial information into your traditional gene cell matrix. And then you can use it for whatever you want to use it for. And we've demonstrated it extensively on cell typing and domain segmentation. But we'll also talk a little bit about batch correction and then some more other tasks that we didn't implement it for, but similar methods that do similar things have done, and we can talk about that. So, okay, so yeah, so two broad problems within cell labeling in spatial omics: there's cell typing and domain segmentation. So cell types, traditionally, you would say cell types are defined by the transcriptome, but we would argue that also the set of neighbors can be influential in at least in influencing the state a cell is in, even if it's not something that you would use to define a cell type, right? And the other thing is spatial domains. You can label domains by the average cell type composition, or you can use the average expression of genes in a given neighborhood. That's the other proxy that you can use to define spatial domains. And we'll go over how Banksy is able to solve these two labeling problems. Right. Before we get into that, there are a bunch of, or when we published the paper, there were a bunch of methods already in the space that try to address these problems. In particular, you could divide them into two broad classes. One is Markov random field-based methods, like there's Giotto's HMRF method, there's spice mix, there's base space. And these methods tend to be quite complex and struggle to scale to very large data sets, like million cell data sets. And there's also deep learning type methods, which will have a lot of weights. They can overfit, unable to scale, with the exception of one method, Stargate, which we managed to get to scale to 500,000 cells. So that was pretty good. I'll show you guys a scaling graph at the end. So, but generally, even the deep learning methods went up to like 200,000 cells or in that ballpark. So, okay, so quick background on what got us thinking about Banksy in the first place. We were just looking at the HMRF methods. And essentially, the idea is that, do I have a mouse? Okay, so the idea is that you want to label a cell using its observed gene expression vector and the majority label, the majority cell type label given to its immediate neighbors, right? The red cells here, whatever cell type label you've given to the majority of these red cells would bias the purple cell to get that label. So you're regularizing the label given to a cell by its neighbors, and that's generally the paradigm within what's called the POTS model in this framework. And so essentially what you're doing is some sort of spatial smoothing or biasing a cell towards its neighbors' labels, and that's useful for something like domain segmentation, but not for cell typing. And we'll go over what we even mean by spatial cell typing a bit later. And the other thing is, it's also a little problematic from an assumptions perspective, because implicit in this framework is that the expression of your index cell is similar to the average expression of the majority cell type class in the immediate neighborhood, which is not really true if you consider like intermingled cell types, in a domain. but despite that slightly not accurate assumption, HMRF type methods do a pretty decent job on domain finding. So we got thinking, can we do something much simpler and still solve this problem? And we thought, okay, what about just taking your gene cell matrix, so the rows here are genes, columns are cells, taking your gene cell matrix and appending some proxy of the neighborhood expression to it, right? So here, for example, the simplest thing you can do is append the average expression of a cell's neighbors to it. And notice we're not adding the average expression of a cell's neighbors to its expression, which is what implicitly a lot of the graph neural networks type methods do, where they do message passing between adjacent cells. But instead, we're appending. So we are lifting a cell from just the gene expression feature space to a feature space that's the Cartesian product of the gene expression and the average expression of the neighbors. And this lifting property will enable cells to retain, to not mix that information and to leverage both pieces of information for cell typing and domain segmentation, as we'll see in a moment. Okay, and in practice, we find that just using mean expression of the neighbors is simple, works all right, but you can get a little bit of a performance boost if you also incorporate the gradients in like other features, for example, the gradient in gene expression around each index cell. So we developed this thing called what we call the azimuthal Gabor filter, which is essentially a Gabor filter, but in the circular azimuthal direction, and that captures a bit more structure in the neighborhood. But you could imagine more generally appending other, like training a variational autoencoder and getting an embedding there and appending that embedding vector to the matrix. So okay, here I'm gonna describe conceptually what's happening, and just bear with me here. This will be useful in understanding why Banksy solves the problems that I'll discuss in a moment, right? So imagine you've got two cell types that are very close together in transcriptome space. So that's what we call own expression space. And essentially, if you lift them to the product space of the own expression and neighborhood expression, you can separate those two cell types out much more. And that can be useful for things like denoising, for example, where let's say I've got two slightly different cell types that are close together in transcriptome space, and normally a cell typing method would label these cells as blue cells and these cells as green cells if all it knows is if all it's looking at is transcriptome, and it would do so rightly because these cells are within the blue region. But we posit that cells, if cells have characteristic neighborhoods, then you can use that neighborhood information to help disambiguate cells that are near decision boundaries. So in particular, this cell, if it's in a neighborhood with all these other cells over here, it should be labeled as blue, and these cells should be labeled as green. And so this is like a hypothesis, and we thought, okay, let's try this out on real data and see if this sort of thinking plays out with real data. So the data we decided to try to use is the RCTD data, which is single cell resolution, but not single cell, because there's a bunch of beads arranged on a chip, and cells need not align with the beads, right? So the data can be quite noisy. Cells of different types can be in a single bead, and so what the RCTD folks, RCTD stands for robust cell type decomposition. What those folks say is that, look, we've got this data set where non-spatial clustering incorrectly calls a bunch of cells that are not granule cells. But so then the RCTD method says that we can use single cell reference data to try to deconvolve this, and they show that yes, indeed, when you use single cell reference data, you get much cleaner cell type labels in slide seed data compared to non-spatial clustering. So we thought, okay, we've got this hypothesis for how Banksy might be used for denoising. Can we do so without single cell reference data? So, because we might not always have reference data for a given data set. And we found that, yes, indeed, we're able to get similar performance to RCTD without using any reference data when we leverage the neighborhood, the fact that cell types tend to have characteristic neighborhoods. And the other use case is of this lifting process is, let's say you have a cell type that resides in two distinct neighborhoods, right? So let's say the blue cell type is at two positions along this vertical axis. So there are cells of the blue cell type which have distinct neighborhoods, and generally cells tend to interact with neighboring cells, with nearby cells. And so you could imagine that the state a cell is in can be different if it's in a different neighborhood. And that can be, and so if you're embedding cells in this product space, that can be useful to distinguish these subpopulations. And indeed, we tried to check this hypothesis on a data set. This is the Moffitt-Murfish mouse hypothalamus data set. And we found that when we looked at the oligodendrocytes, there were mature oligodendrocytes in the anterior commissure region, which is a white matter region. And there are oligodendrocytes within the general hypothalamic area, so within the gray matter. And Banksy separated these two oligodendrocyte populations out. And we found that there were a bunch of genes that were differentially expressed between them. And the genes that were upregulated within the general hypothalamic area were enriched for, or these cells were enriched for genes that were involved in signaling, whereas the genes enriched here were more to do with myelination and so on. And you can also plot own expression metagene versus neighborhood expression metagene corresponding to these genes. And you can see that this situation that we showed in the schematic in our hypothesis does show up, where you have the sparse oligodendrocytes, the gray matter oligodendrocytes versus the dense oligodendrocytes, even though in the own expression axis, they're highly overlapping and therefore very difficult to distinguish via non-spatial clustering. Using Banksy, they're much better separated in this joint space, and therefore Banksy is able to separate them out. Okay, so the third use case is there's this parameter lambda within the Banksy matrix, which Joseph will talk about more during his talk. But basically, the idea is that we weight the own expression and the neighborhood expression for each cell by this lambda parameter. So if lambda is, let's say, 1, what you get is this term is 0. So you multiply 0 into the own expression, and you multiply 1 into the neighborhood expression. So essentially, your feature object matrix is your features are just average neighborhood expression for each cell. So essentially, you're clustering by the average neighborhood expression, and so it becomes a domain segmentation method. Whereas when lambda is 0, this term is these set of features are ignored, and so you're only clustering by own expression, and this reduces to standard non-spatial Seurat clustering, right? Or the standard non-spatial clustering. So, okay, so in domain segmentation, when lambda is close to 1, you're essentially clustering by average neighborhood gene expression or average cell type composition. And you end up with, earlier we were looking at the hypothalamus data set in cell typing mode. This is the same data set in domain segmentation mode. And you can see like the bed nuclei of the stria terminalis, the medial preoptic area, the third ventricle, and all of these regions sort of coming out as domains. And also like the DLPFC data set that everybody uses for benchmarking. Our approach does pretty well. It's sort of on par with the state of the art in the field, so on. It's all within the same framework. And it was nice to see that this is on the Seurat website. They have a spatial framework now, and they suggested using Banksy for Visium HD data analysis. So we have a Seurat wrappers version of Banksy that you can use with Seurat objects. Although today, Joseph will just show you the Bioconductor SingleCellExperiment spatial experiment framework. But yeah, you can use it with Seurat, or if you prefer Python, it's also in the ScanPy framework. Okay, yeah, and the other use case is batch correction. So this is sort of going a bit more general than just cell typing and domain segmentation. Basically, the idea is that it's a feature object matrix that has spatial information incorporated into it. So you can just use it for whatever you would use your gene cell matrix for. In particular, if you wanted to run harmony, you would take your gene cell matrix, compute PCA scores, and run harmony on that. So same thing, you can take your Banksy matrix, compute PC scores, and run harmony. And we found that that's a really good approach for data integration, for batch correction. And yeah, it does well across a bunch of different metrics on data sets that we have ground truths for. Yeah, so on scalability, this is becoming more important as the data set sizes increase, I guess. So like I mentioned, Stargate was a deep learning method that scales really well, but the other methods do tend to sit on the 250K-ish data set size, some of them even lower than that. Banksy is able to scale to 2 million or more because it's basically not doing anything crazy like HMRFs or deep learning, right? You're just appending a bunch of features. So you're just, I would say in a very simple framework, you're incorporating spatial information, but still solving the same problem. So you can just leverage lead-in clustering or even k-means clustering if you wanted to, and just go to like a very large data set size. There's also like this package called BP cells that's somewhat recent-ish, and you can use that to scale up even higher, like 4 or 5 million cells if you wanted to. So really, it basically scales with the state of the art of like graph-based clustering. And oh yeah, and there was this paper by Shamir and co. where they showed that there's no reason why we have to use this framework for only genomic data analysis or transcriptomic data analysis. So they said, we want to solve this problem of sort of parceling out the regions on the surface of the brain, cortical regions on the surface of the brain, and our features are layer thickness within the cortical layer thicknesses. And we're just going to use Banksy with lambda 0.8, a high lambda value in domain segmentation mode, and try to just segment out MRI data. And they showed that that works well and does better than existing methods. So that was very cool to see. And yeah, so that's the conceptual side of things. I'm very happy to take questions. I can take them now. I can take them after Joseph's section. But yeah, I want to thank Nigel, who was my co-first author on this, and Joseph, who wrote a lot of the code, and PIs, Kakao, Sham at GIS, and Hui Guan at the Bioinformatics Institute in Singapore. And thank you.
All right, yeah, so I'll be walking through 2 demos today, just illustrating some of the concepts that Vipul went through in terms of like parameter selection and especially multi-sample analysis and batch correction. So this is available on the workshop, so you should be able to spin up a server if you want to follow along. It's also on GitHub, and you can probably refer to the main package repository for like our vignettes and everything. Yeah, but I'll just demo maybe a standard workflow and some of the parameter choices that are relevant, I guess. So yeah, we'll be working with a data set that's attached with the Banksy package. So it's a hippocampus data set comprising about 10K cells across 120 genes. So it's, I guess, pretty small in today's standards. Yeah, I'm just going to load some packages here. The data looks like this. So this should be pretty familiar to everyone as well. So just a gene cell matrix and then a matrix of locations corresponding to the cell centroids. And then we create our spatial experiment. Yeah, the spatial experiment, I guess, has this spatial cohorts, I guess, slot that builds on the SCE. So that's what we use to extract the cell centroids, basically. And yeah, we perform basic quality control, just filtering by the total count, so between 5% and 98th percentile of the whole data set. So that leaves about 10K cells. It's pretty simple. And then do standard normalization, so a log 1p. Yep. Sure. Is this big enough? Okay. Yeah. Okay. No worries. get this out of the way. Yep, okay, so normalization is done. And then this is where I guess the main stuff is at. So the first thing is AGF usage. So as Vipul said just now, you have the own expression matrix. You also have the neighborhood mean average expression. And then the third chunk of the matrix is sort of the gene expression gradient in the neighborhood. So we've noticed like in practice, whether you include the AGF or not, it seems to give a slight performance boost, but it also increases memory usage. So yeah, here you can determine whether you want to use that feature. So if you just set compute AGF, we have a simple Boolean flag that says whether you want to compute the mean expression and the azimuthal Gabor filter. The second, I guess, important parameter is k-geom. So this determines the size of your neighborhood. So we found that in practice, 15 to 30 works well. In like domain-specific applications, for example, if you're looking at Visium, where there's like grid-like structure, you may want to consider, for example, first-order neighbors, which correspond to 6 neighbors, and then first and second-order neighbors, which correspond to 6 plus 12, so that's 18. Yeah, so I guess those are two important parameters that you can set. And another thing is we also allow like different neighborhoods for different chunks of the matrix. So we found that to compute gradients, especially because those are differences in the neighborhood, using a larger neighborhood helps to sort of reduce the variance in that component. Yep. So I guess by default, Banksy uses 15 for the average expression. So that's the first set of augmented features. And then for the second set of augmented features, it's twice that value. Yep. So here is the compute Banksy function. So what it's doing is first, computing the neighbors for each cell, and then computing basically the average expression here. So that's harmonic m equals to 0, and then the azimuthal Gabor filter, which is m equals to 1. And you can see the number of neighbors it's using here. So if you look at the spatial experiment object now, it has the counts, the norm counts, and then it adds 2 assays. So the mean expression and then the azimuthal Gabor filter. Yep. So as Vipul was describing this, depending on your task, whether it's cell typing or domain segmentation, you can choose the parameter lambda and adjust for that basically. So it's between 0 and 1. And by default, we use 0.2 for cell typing and 0.8 for domain segmentation. Yep. So a point to note is that in certain like high resolution assays like Visium, for example, where it's not single cell resolution, you might want to use a small lambda for domain segmentation, even though it's lambda 0.2, because the spot already contains multiple cell types. Just use a small lambda so that you don't average out even more signal. Yep. So here we're running with both lambda 0 and lambda 0.2. So lambda 0 basically corresponds to non-spatial clustering, where you zero out both the average and the Gabor filter. Yep. So what this does is basically runs PCA. So it takes the chunks of matrix and then basically computes the principal components on that. So by default, we use 20 PCs. Yeah, but of course, you can change that. And yeah, the same thing with UMAP, I guess. So basically, this is telling the function to use the AGF and then run this on both these lambdas. Yeah. So hopefully it doesn't take too long. Yep, go ahead. Yeah. Maybe we can handle that at the end. Yeah, I think that would be good. Right.
I think you can answer that question very generally. Like, have imaging data, you could convert that into, you could directly use the pixel values, I guess, the channel values, but you could also use some sort of autoencoder type thing to embed that data in some sort of embedding space and append that as a set of features, right?
Yep.
Like we've not tried that, but I think that's sort of the direction we're thinking of going. Actually, two directions. One is using the Banksy matrix as an input into existing deep learning spatial frameworks and just seeing if we can improve things that way, or taking the embeddings learned by existing deep learning frameworks, including image type embeddings, not genomic data set type image data set, taking those embeddings and appending those to the Banksy. So both directions, feeding the Banksy matrix in or constructing a non-genomic informed Banksy matrix. Right? Yeah. And also those multimodal, right?
Well, actually, just while you're on that, said yeah, histology and multiplex immunofluorescence.
Yeah, I think so. This is that matched unmatched thing comes into this. So, for example, as long as for a given cell you have those features, like you've got your IF data, you've got your histology, then I think pre-processing is usually the key there. Like you do want to get that into some sort of compatible state, but probably the first place to start is use standard pre-processing steps that exist for those data sets and just get those, just append those values and see how that goes. And multimodal, the same answer, I guess it needs to be that match. Cell should have those.
Okay, because they say you have multiple aligned data sets that are single cell.
Aligned data sets that are single cell. Okay, aligned, so I'm guessing means like using integration methods to align cells that were separately measured. I'm not sure. I think maybe more about. Yeah, I don't want to say something else. Right. So.
Okay. Yep. So that's done. You can see the reduced DIMS slot now contains the reduction, so PCAs for 0 and 0.2, and then UMAPs for 0 and 0.2. We then go on to clustering. So this just now works with in terms of the PCs. So we implement a variety of clustering methods. So we have Leiden, Louvain, K-means, and also M-class. By default, we use Leiden. But of course, if you have you're looking for a fixed number of classes, you can use K-means, for instance. So I'll just run this at 2 resolutions, so 0.5 and 1, and then with a K expression of 50. So this tells you how to construct the graph, basically, in transcriptomic space that we then cluster with the Leiden algorithm. Yep. So once clustering is done, the cluster labels are stored in the call data of the object. So you can see the corresponding label. So it's lambda 0, K expression of 50, and then a resolution parameter. And then we also implement a function to connect different cluster labels. So I think it's a problem that we face in visualizing results in particular. So if you want to compare clustering runs, the numbers, the literal numbers may not be aligned between the clustering runs. So we implemented a simple function based on Hungarian matching algorithm to just connect the different labels from different clustering runs. So this is how the connection runs. And then basically we do some simple visualization using plot call data from Skater, I think. So yeah, what you can see here is basically the non-spatial on the top at different resolutions. So I guess there are 2 more classes here, and then at a slightly higher lambda, where you do spatially informed cell typing, you get also, it's a bit hard to tell the difference in the slide, but you can also wrap it by the clusters basically and see how by incorporating that 0.2 lambda, the classes change slightly. Yeah, so I think we did this in the paper, and you can refer to the paper for the figure, the exact figure. Yeah. So I won't go through the UMAPs. We also implement a cluster comparison function. So for example, if you have a ground truth annotation and you want to compare your cluster labels to the ground truth annotation, we implement using the package recode. It takes in different metrics. So for example, ARI is pretty popular, I guess, in the field. And also, for example, NMI, normalized mutual information. So you can see how the classes compare. So this is a pairwise comparison between the different clustering runs. And then for NMI, yeah. So think it's pretty handy. Last but not least, I guess, is if you're finding smooth domains instead. As Vipul said, you can use a high lambda to outweigh the neighborhood augmented features versus the own expression. So I'll just run this whole chunk, which basically repeats the analysis above, but just with 0.8 lambda. Hopefully it won't take too long as well. Yeah, so in practice, I guess this use AGF thing, you can, if you want to include it or not include it, you can also just add another term here like false, for instance. So that'll iterate through also using the AGF and not using the AGF. So it's pretty general in that sense. The docs will probably be a good place to explore like which parameters you can provide like vector arguments for that the functions will iterate through. So PC is done, and then we run clustering with the 2 lambdas at fixed resolution and then 50 expression neighbors. So if you compare it to the previous lambda 0 and lambda 0.2, you see that the domains are smoother, I guess, for at least at high lambda. So if that's the sort of biological question that you're trying to target, like segmenting domains in your data set, then this might be useful. Yeah. So now I'll move on to multi-sample analysis. I guess this is also pretty popular nowadays, where you have like multiple samples from the same tissue, for example, and you're trying to integrate them. Yeah, so I think I've already loaded data, and it's basically the DLPFC dataset. The original dataset has 12 slices from 3 different subjects. So here what I'm going to demonstrate is taking one slice from each of those subjects and then integrating them. Yeah. So this is the SP object, which has already the data from the spatial LIBD package. And I'm just going to trim it to save memory, basically. Yeah. So this is just pre-processing, and I'm extracting the samples from corresponding to 3 subjects here. And then in their annotation, so they have pathology annotations, which is why it's used in benchmarking. We're taking the white matter and the 6 cortical layers, and then leaving out a sort of cluster that they didn't annotate. Yeah. So that does that. And then, so for multiple sample analysis, typically in your objects, you have sort of this scenario where I'm visualizing the slices here, but the coordinates from different samples overlap. So when we're computing the Banksy matrix, what we want to do is compute the neighbors within each sample separately. So the colors here are the samples, basically. And I've written here a simple function that does this. So this is not incorporated into the package, but I think we should do that yeah, because I think it's useful. So this is how the steering looks like. So basically, it shifts each subject's slice in the horizontal direction. So when you compute the Banksy matrix, it just pulls neighbors from its own sample rather than across the slices as well. Same thing we're going to do, just normalization here. I'm taking some highly behavioral genes because it's Visium. So I think the original data set was like 20,000 features. And for this data set in particular, we found that just using count-based normalization, so normalizing by the library size works well. Somehow using logs leads to like poor performance downstream. Yeah, so, and the same thing again. In this case, because it's Visium, we're using first order and second order neighbors. So that's 6 plus 12, 18 neighbors to compute the features, the augmented features in our data set. And we're using also the azimuthal Gabor filter. And then again, running Banksy with non-spatial and then a 0.2 because it's Visium. And we don't want to use a 0.8 to over smooth our data. Yeah. So I guess in a typical analysis, the longer steps are usually the PCA and the clustering. So PCA is compressing that neighbor augmented matrix into PCs. So that's where the bulk of it is. Once you have the principal components, then the downstream, like computing UMaps is just on, for example, 20 PCs. So yeah, it's independent of the number of features afterwards, after this set basically. But yeah, the Banksy matrix computation is linear in the number of features. So as the feature set size grows, then you basically double, for example. And we're thinking of strategies to sort of make it more efficient as the data sets grow larger in terms of the number of features, especially. Yeah. So if you have any feedback or ideas, we'd be happy to take them as well. Yeah, another thing we found is that sometimes if you want to do multi-sample analysis, it's also just possible to z-scale your samples separately, and that sort of gets rid of minor batch effects or like accounts for those. But if you have very differing samples from different subjects, for instance, then using tools like Harmony may help. Yeah, so in this data set, for example, if you're looking at 4 slices from the same subject, then just doing that z-scaling trick might work. Yeah. But from different subjects, then using a tool like Harmony use sort of better performance. Yep. So once the PCs are computed, so basically you can think of these as sort of spatially informed embedding of your data. And then we basically run Harmony on the non-spatial PCA and also the Banksy PCA here. So once that's done, we add those embeddings, the Harmony embeddings back into the spatial experiment object. Yep. And then we just basically do clustering on the embedding. So here I'm extracting the PCs and the Harmony corrected embeddings, and then using k-means clustering because we know the expected number of domains in our dataset. And then we're going to connect our cluster labels to basically the ground truth annotation here. And then basically what you can do is use the same function as before to compare the clustering runs. So you can see this is the annotation to itself. That's why it's 1. And then you have the non-spatial, non-corrected, the spatial, non-corrected, the non-spatial corrected, and then the spatial corrected. So the numbers are not that informative here, I guess. It would be helpful to look at visualizations of the data. So here's the ground truth annotation. You have the white matter and then the 6 cortical layers. So this is subject 1, 2, and 3. So this is the non-spatial result before correction. So it's basically noisy. You get like sort of noisy classes and they don't really correspond to smooth domains. This is Banksy without correction. So you can see that you sort of get nice classes as well, but it's not as good performance, I suppose. Yeah, I guess you sort of also get cluster-specific samples sometimes. That's what we see when we are merging different data sets from different subjects. This is non-spatial with Harmony. So again, noisy. And then Banksy with Harmony. So yeah, in this case, the domains are, I guess, smoother. Yeah, but then our benchmarking in the paper, basically we run it on each data set. That also helps, I guess. Yeah, so that's all for the demo, and happy to take questions about the method and also the code. And if you have any suggestions on like the user interface and experience, that would be very helpful. Yeah, thank you.
Hi, thanks for this demo. My question is a bit more conceptual with Banksy. And this is an assumption that underlies a lot of spatial analysis. And that it's the assumption that the nearest neighbors physically are going to be very important to the kind of expression profile of the cell. And while that's true in a lot of cell cases, and in neurons biologically, that's really not the case because they project. So have you actually tested whether or not the best cluster or the best explanation of the covariate structure is actually the nearest neighbors? And further, can you change the niche to a distal location from the actual location of the cell using Banksy?
So that's a really great question.
Speaking of Mexico, online, hear the answer.
Yeah, that's a really great question. Short answer is no, we've not tried that. I think even conceptually, so we have a lot of different spatial kernels that we've implemented as possible arguments within the method, but none of those kernels have that annular shape that you might imagine might start to move in the direction of answering your question. You know, it's like it rejects cells that are nearby, but then takes cells that are further away. We haven't done that yet, but I think that sounds, I don't know. But then again, annular shape is such a simplistic idea, right? Maybe something more complex than that. I don't know, like maybe like a graph transformer type thing where some attention mechanism attends to the right neighbors far enough away, being the things that inform neighborhood structure. I'm not sure. If I had to guess, that would probably be the first thing to try to develop things in that direction. That's a much harder problem than just taking nearest neighbors, what we're doing, right? Yeah. Like figuring out which neurons affect a given neuron. Yeah. And
because like specific neuron cell subtypes have known projection patterns, right? So like layer 5 IT neurons in the cortex project to the striatum.
Yeah.
Right? So that you can actually use ground truth knowledge to restrict the search space.
Yeah.
And you can even look as simply as just covariate structure for the expression pattern overall.
Yeah.
Right?
Yeah. Yeah.
But
yeah,
would it be possible to do that with Banksy to actually change actually manually set the niche?
I think it would need some development work. There's nothing conceptually that's stopping us,
yeah.
but that function needs to be written, yeah. Right? That function where you say, okay, I want some method of defining my neighborhoods that's sort of more complex than nearest neighbors or Delaunay or whatever. And yeah, I'm not sure. I think, how would you do that? Like, how it sounds like some expert knowledge is needed there, right? Like what you just described, this layer interacts with this layer.
Yeah.
And for every layer, it's a different layer, right? So then doing that automatically is hard. Yeah. Maybe the attention type methods could help there, but I'm not sure. Yeah.
Cool. Thanks.
Yeah. Thanks. Yeah, sure. So quite a few data sets. I'll just go through the figures in my head and list out the data sets, I guess. For cell type, for testing cell typing capabilities, we use the SlideSeq data set for denoising that we talked about in this talk, both SlideSeq version 1 and version 2, for showing that a single cell type can exist in distinct neighborhoods. We used the Murfish mouse hypothalamus dataset and the CRC. There's a colorectal cancer dataset by Merscope that's also in the paper. So that's figure 3 if you're looking for it. Figure 4 was a Verafish dataset that was collected by some collaborators of ours. That was also a single cell resolution fish-based dataset, very similar to Murfish. And that was also a cell type test where we showed that oligodendrocytes in different neighborhoods had subtly different expression and neurons in the somatosensory cortex versus the pyramidal neurons in the CA3 region, where even though they were the same cell type, they were subtly different because they were in different regions. And then moving to domain segmentation, there was the StarMap dataset, the mouse visual cortex, I believe. That had ground truth labels, so there was some ARI calculations there, right? You can compare to other methods and so on. There was also the DLPFC data set. Again, everybody uses that for benchmarking. There was simulated data sets that we generated using SC Design 2, and there we were able to also do some benchmarking. And I think I've gotten all of them. Have I gotten all of them? Yeah, I think that's it. Yeah, so like 7 or 8 or 9, something like that. Yeah. Cool, thank you.
Thanks for joining this tutorial workshop. We do have a break now for almost 20 minutes. There are snacks outside, so they should be putting snacks out there. In about 5 minutes, they're also trying to grab everybody in the main atrium for a big photo for the conference. If you're keen on that, go to that. If you're not comfortable being photographed, filmed, shot, whatever the word I'm looking for is, don't go to that. You're fine. No one will judge you. See you out there. Enjoy the snacks.